bell is rung. It's time to go. Hello, freshman honor students. I have truly missed all of you, and I'm totally serious. I hope all is well. Use this time productively. This is a very good time to enhance your intellectual interest. And now that I've got some things out of the way, ready to go, got all the little glitches out of the way, I'm going to help you achieve those goals, whether you like it or not. Actually, just kidding there. So please use this time productively and pursue intellectual interests. This is a golden opportunity. Anyway, again, welcome back. And let's see, we were talking evolution, Darwin's theory of natural selection. That natural selection process is relying upon mutations, favorable mutations. If a living creature has a favorable mutation, they're going to have a greater chance of survival. Therefore, they may live long enough to reproduce and pass on that favorable mutation to the next generation. So it's mutations. Favorable mutations are the engine that drives the evolutionary process. Of course, we also know that these mutations can be harmful. We have excellent projects, and here's one right close to me. Mr. Noble, Huntington's disease. I mean, this is excellent, and I kept these projects that you guys have done. They really are good, but these diseases are examples where mutations are resulting in a disease of some sort. And of course, we could review maybe a little bit all the different types of mutations that we've talked about. There's substitution mutations where a DNA base maybe the A is substituted in place of a T, for example. There's frame shift mutations where a DNA base is removed or added, and it's going to throw off the reading of the genetic code. So each codon from the point of the additional new base is going to be off. And then every frame after that, every set of 3 after that is going to be messed up. These can have profound effects on proteins and therefore effect on living creatures. So let's keep some of that in mind because it is going to be important as we move forward when we talk about viruses and bacteria, because viruses have mutation rates, they have change rates. The DNA or the RNA of the virus will alter from one generation to the other. This is what makes fighting viruses like the flu so difficult to create vaccinations for, because of the mutation rate. The offspring aren't exactly genetically identical to the original viral components. A lot of this we're going to talk about in detail because it's timely and really, really, really important. Well, the next thing we did after we talked about mutation, natural selection, and we also discussed evidence
Mrs.
for
Sands,
evolution.
this is just a reminder, you have yoga class in a couple of minutes. Mrs. Sands, you have yoga class in a couple of minutes.
I gotta see this. Just kidding. We've got to pursue our intellectual interests here. Anyway, where was I? So we talked about evidence for evolution, and I want you to review this. It's been 2, 3, 4, 5 days, so go back in your notes and review that information. Let's keep some things in our head. The next thing we did, we've got this tree of life where there's unity of life. life evolved from a common ancestor, and that tree of life is branching off into different categories of organisms. This is classification, or the categorization of different living creatures. The science of classification is, of course, taxonomy. Well, currently, there's a 3-domain system of organization for living creatures. 3-domain. All a domain is, it's a large category, has a lot of living creatures in it. But keep in mind, these categories are not just hodgepodge random. They are based on genetic and structural similarity of those living organisms that are in each of those domains. So we have bacteria, we have archaea, we have eukarya. Those are the 3 domains of living creatures. Those domains can be broken down into kingdoms. So we have a 6-kingdom system of classification. So the domains are the broadest categories. They have the most living creatures. But the domains can be broken down into separate categories called kingdoms. What are the other kingdoms? We have eubacteria, we have archaeobacteria. These are prokaryotic, primitive living creatures. When I say primitive, you need to keep in mind that even a primitive microorganism is very complex in its cellular machinery and in its metabolism and in its reproduction. So even though I say primitive, remember that life, if it's still here, it is working really efficiently, and it is very, very, very complex. So these so-called primitive microorganisms, which would include the eubacteria and also the archaeobacteria, they're prokaryotic, so they lack membrane-bound organelles. They lack, what else? They lack a traditional nucleus. They do have genetic material. They have small plasmids, which of course we use in genetic engineering because we can engineer those plasmids and add genes from other living creatures. If I may review a little bit, let's take a plasmid from a bacteria. Let's open that plasmid up with restriction enzymes. Let's add a new gene. Let me just grab something here. Let's add a new gene. So notice this represents a new gene that we've added, and this gene could be a human gene. Then we're going to give this back to the microorganism. Hopefully they'll take it in, and we will, assuming they've taken in the new plasmid, the recombinant plasmid, they will be transformed, and this is called transformation. And once they start reproducing, and when they reproduce, they're going to reproduce exponentially. Now that's important to understand, and I'm going to talk about that in another video, but this idea of reproducing exponentially is really important in how populations grow, whether it be insect populations, whether it be bacterial populations, and yes, whether it means infection due to, well, let me go back here, whether it means viral reproduction can reproduce exponentially, and infections of individuals can occur in an exponential progression. So we're going to go back to that in a little bit, maybe in a different video, which is probably going to happen today, that idea of exponential growth. Anyway, once we have that large population of bacteria which have taken in that recombinant plasmid, the plasmid that has the human gene in it, then we've got this large population because they're reproducing, and then of course they'll start transcribing and translating that information into their own proteins, but then also into the new protein that was produced by the human gene. So insulin, for example, interferon, factor VIII, which is an important medication for hemophilia, and many other medically important therapies have been produced using this process. Anyway, so we've got those eubacteria, prokaryotic. We also have the archaeobacteria, and these bacteria are often called extremophiles. Many have the ability to live in extreme environments. And what are some extreme environments? Try water temperatures of 150 degrees Fahrenheit. It's amazing. There's bacteria that can live deep beneath the ocean surface where pressures are tremendous. You can get pressures that exceed many tons per square inch the farther you go down from the ocean surface. These bacteria will thrive and survive around volcanic vents, and of course there's no sunlight there. So they are getting energy from a process called chemotherapy. So they're using the chemical products at hand, sulfur compounds that are close by, and they're metabolizing those in a way that releases energy. So they're not using light to produce energy, they're using the energy of these sulfur compounds. So these archaeobacteria are interesting in a lot of ways. Scientists do think that they are, in fact, the precursors to modern eukaryotic cells, those more complex cells that have arisen more recently from an evolutionary standpoint. Is there evidence for that theory? Is there evidence that these archaeobacteria have led to eukaryotic cells and therefore more complex multicellular creatures like plants and animals? Well, yeah, there is. It seems that the archaeobacteria they use introns and exons just like we do. So we've talked about introns and exons in gene expression. A typical bacteria would not use that same type of arrangement of the DNA with the introns and exons. Also, the DNA of the archaeobacteria is wound with histone proteins. These histone proteins are similar to histone proteins that our DNA and eukaryotic organisms have, that those histone proteins help to organize the DNA. So the archaeobacteria have them, but the eubacteria don't. Well, we go from those primitive microorganisms, so-called primitive microorganisms, to eukaryotic organisms. These can be single cellular creatures or multicellular creatures. They have a complex cellular structure. They have a division of labor for the organelles. This division of labor are membrane-bound organelles, so we're going to find mitochondria. So division of labor means we've got this little factory here doing this thing, this little factory here does something different. All of these little organelle-like factories are doing their own thing to help maintain life. So we could have the mitochondria being the powerhouse. We can have packaging centers. We can have transport centers. Different organelles that are involved in reproduction. That would be centrioles. Anyway, what are the kingdoms in the domain eukarya? We have protista. We have fungi. We have the kingdom of plants, and we also have the kingdom of animals. Most of these are going to be multicellular living creatures, but not all. They all are going to have their own unique characteristics that make them slightly different from each other. Of course, they're going to be similar to each other in that they all have membrane-bound organelles and a nucleus. Well, what I would like you to do now is just review the characteristics of all the different living creatures in the six kingdom system of classification. This has to do with your last assignment. This is meticulous. You just need to look at it again, look at it again. So go ahead and do that. All right, so we're back. I hope you reviewed some of the taxonomic categories and their particular characteristics. So, are you ready here? Which kingdom? You ready? Call out here, unless somebody's sleeping. If you have a little brother or sister that's really young, don't call out loud enough to wake them up. We don't want to mess with anything here. So, get ready to call out. All right, what kingdom? Remember, I'm in a kingdom now, I'm not in a domain, so we got to limit our discussion to kingdoms. What kingdom has a cell wall of chitin? William. Louder. Did you forget that? Chitin? Chitin is a particular type of cell wall, and that cell wall is made up of a carbohydrate. We've talked about carbohydrates. Let's see, what were they? The complex carbohydrates: starch, glycogen, cellulose. Chitin is a complex carbohydrate, and that makes up the cell wall of fungi like mushrooms. All right, what living creature has a cell wall with cellulose? Who is it? Anybody? Anybody? Bueller? Bueller? It is... could be protist, but more commonly it's going to be plants. Which of the prokaryotic living organisms has a cell wall which lacks peptidoglycan? Look that up. Which prokaryotic living organism has a cell wall which has peptidoglycan? Look that up. What is peptidoglycan? Think of it as a part carbohydrate, part protein-like cell wall. So that's peptidoglycan. Escherichia coli, E. coli, is in what kingdom? Look it up. Paramecium caudatum, or simply paramecium or amoeba, are in what kingdom? Go ahead and find that. Yeast are in what kingdom? Go ahead and find that. Well, we've talked kingdoms. Well, we've talked domains. Now we're talking kingdoms. So we have eubacteria, we have archaeobacteria, we have kingdom protista, the fungi, animals, and plants. We can break kingdoms down into smaller and smaller categories. So the taxonomic classification of kingdoms can be broken down, with kingdoms being having a lot of living creatures in it, and then each successive group will have fewer and fewer living things till we finally get to the genus and the species of one living creature. So take a look here. This is, we've got a kingdom, and we could have, let's say, the kingdom of animals, of which we are a part. Of course, that could be the kingdom of fungi, or it could be the kingdom of eubacteria. Now, the next category, and a phylum here, a phylum is simply a category. That's all it is. And the category of the phylum is going to be a broad category. The class is going to be less broad. The order is going to have fewer living creatures in it. Families have yet fewer living creatures. Then finally we get down to the genus and the species. Anyway, let's go through us. We've got kingdom of animals. We're in the phylum Chordata. That phylum includes most of the animals that you're familiar with, whether it's a dog or cat or a rabbit, or even invertebrates like earthworms, for example, or sponges. So most of the living creatures that you're familiar with are going to be in this phylum, bacteria. And then the next taxonomic category is phylum. Let's go through this for ourselves. Let's go through this for us. So we are, of course, in the kingdom of animals, or Animalia. We'll use the Latin term. We are in the phylum, and the phylum, again, is just a category. That's all it is. It's a category, and the living things in this phylum have certain characteristics. For example, they're developing in similar ways embryonically speaking. And then you can still get some animals that are kind of outliers in the kingdom of animals, which have some interesting and important differences in how they develop embryonically compared to us. But they're still animals though. Anyway, most animals are going to be in the phylum that we belong to. And in fact, invertebrates, most invertebrates I should say, are going to be in the same phylum that we belong to. Remember, the higher up you are, the larger the category is, and the more living creatures are present. So let's see here. So we're in the kingdom Animalia. We're in the phylum Chordata. That's our phylum. We're in the class of Mammalia, or mammals. So we're in the kingdom of animals, the phylum Chordata, the class Mammalia. What order do we belong? We're in primates. What family do we belong? It's called, we're hominids or hominidae. So hominidae, hominids. What genus do we belong to? Homo. And what species do we belong to? We are sapiens. So oftentimes we will define an individual stating their genus and their species. So here we are Homo and sapiens. And let's underline that. This should be, if you were typing this out, it should be in italics. You should have the genus here, capital, and you should have the lowercase for the species. So that should be lowercase. Again, when you read this in your text, it's going to be capital because it's a scientific name. So we are Homo sapiens. We could look at the mammals. There's lots of mammals that you're familiar with. Mammals have certain characteristics. Hair on the body, going to be air breathing, going to give birth to live young, so there's no shells involved here. We are, I take that back, there are a few monotremers, there are a few different animals that are egg-laying mammals, but in general we're going to give birth to live young. And here we are with the genus and the species. This is us, Homo sapiens. Notice this should be italics, that's why it's underlined. We're going to capitalize the genus, lowercase for the species. A white pine tree. Obviously a white pine tree is going to be in a completely different kingdom, obviously the phylum, etc. But once you get down to the scientific name, that would be Pinus strobus. So notice, genus and species. Well, let's see here. It's important to come up with little ways to help you remember things. They'll do this in medical school. Come up with sentences to help you remember the order of a series of words, for example. Be creative. Anything that works is going to be useful in helping you remember material. So, notice here are the taxonomic categories, beginning with kingdoms. So let's underline K, P, C, O, F, G, S. So we're going to underline that. And let's use a traditional sentence to help us remember the order of these words. Again, the order is important because if you're talking about a class of living creatures, we know that this class has more living creatures in it than an order does, because again, you're going from the broadest category to smaller, smaller, smaller. So we'll do the classic. The classic, I learned it when I was your age. Not in this building, in the other building. One of the reasons I kind of miss the other building, but I love this place too. Anyway, it is King Philip came over for grain stamps. King Philip came over for grain stamps. Write it. What did King Philip come over for? Grain stamps. Well, what you can do I'm not going to give you this one. I've got my own little memory device, mnemonic device for this. But what you can do is come up with your own little sentence that begins with A, then the next word is C, M, P, H, H, and S. Write it, and it's amazing how effective these sort of things can be. All right, so we're moving on. Taxonomy. Boom. Kicked it out. It's a little tedious, but what I want to emphasize here is that these categories are evolutionary based. So if they're in the same kingdom, they're evolutionary. All the living things in that particular kingdom are evolutionary, more closely related to each other. Also, these categories are based on genetic similarities. So if you're in the same class as another living creature, so let's say a whale or a bat or a human or a dog, we are genetically more closely related to each other than we would be with a living creature that's not in that class. For example, birds are in the class Aves, A-V-E-S. So we are not as closely related to a bird, genetically speaking, as we would be to living creatures in our class, in the class of Mammalia or mammals. So we're moving forward. This is really interesting stuff we're going to be doing given the series of circumstances going on in the country and along the world, and throughout the world. So we are delving into viruses, we're delving into bacteria. We're going to study fungi slightly, a couple days on fungi. Then we're going deep into the animal kingdom. It's going to be all about animals. And the vast majority of the fourth quarter will be studying animals. We're going to study vertebrates like sponges. We're going to study, invertebrates like sponges, invertebrates like worms or coral. Then we're going to study vertebrates. This typically would be a time where we move into dissections. And hopefully you'll be back then, because it's it's a fun part of the course, and it's something that kind of you get more out of it if you're actually doing it as opposed to me watching it do it on a video. We would be dissecting fish, frogs, worms, the cow-eye, one of my personal favorites, and usually a few other creatures as well, depending on the specific situation. So again, I miss all you guys, girls, everyone, and stay safe out there. I am going to be posting another video, and that's going to be the start of viruses and bacteria. Have a good day.